Passive transport across the plasma membrane means that the substance does not need any energy in the form of ATP to help it to cross the membrane. So passive means no energy investment or no energy requirement. And it also means that the molecule or the substance is going to move from where its concentration is high to where it is low. Diffusion is a type of passive transport. So the molecules that diffuse directly through the phospholipid bilayer are the ones like oxygen and carbon dioxide that are nonpolar and they are small. So diffusion is the movement of particles so that they spread out evenly into the available space. And what that means is that they're going to move from where their concentration is high to where their concentration is low. And you can see the molecules are moving across this membrane. They are moving from high to low concentration until they reach equilibrium. There's a video on diffusion in section 7.3. When molecules move from where their concentration is high to where it's low, we say that they are diffusing down or with their concentration gradient. So for example, these yellow molecules, if they're going to diffuse down their concentration gradient, they're going to move from the left side to the right side. And the purple molecules are going to diffuse from the right side to the left if they're going to go down or with their concentration gradient. And we already said passive transport is when there's no energy requirement. Specific type of diffusion, the diffusion of water, is called osmosis. And what happens there is you have a membrane which is not permeable to certain solutes. But like in this particular, we call this structure a U-tube. We use these in lab to show diffusion and to show transport of substances through a membrane. So in this case, we're using it to show osmosis because you can see that on the left side you have fewer sugar molecules and on the right side you have more. So water is always going to follow solute. It's always going to move toward the higher solute concentration. And the reason is because water moves. Water is like anything else. Water moves from where water concentration is high to where water concentration is low. And you can see a lot more water on the left than on the right. But I just always remember that water follows solute. That helps me remember where water is going to go because the water is going to move to the higher solute concentration. So you can see that the water is going to move toward the right, and it does, and the level of water rises. And what that does is it evens out the concentration of solute molecules on both sides of that U-tube. There's a video that shows that as well. Now, the word tonicity is related to the concentration of solute. So we say that a solution is isotonic if its solute concentration matches the solute concentration inside the cell. So isotonic means equal or the same. And even though if you place a cell in an isotonic solution, there's going to be movement of water into and out of the cell, but it's going to be the same amount. So the cell itself is not going to change. The volume is going to remain stable. The cell size will not change. So the volume of the cell is going to remain stable. If the cell is placed in a hypertonic solution, that means the solute concentration is higher or greater than inside the cell. This would be like a really salty solution or a really high concentration of glucose solution. And what's going to happen is water is going to move toward the solute, toward the higher solute concentration. So water will leave the cell and it will shrivel and probably shrink, and it will probably die. And then if it is placed in a hypotonic solution, that means that the solution has a lower solute concentration than inside the cell. For example, if you place a cell into distilled water, which has no solute in it, what's going to happen there is that water is going to move into the cell because water will follow solute. It will move toward the higher solute concentration. So the cell will gain water, it will swell, and it will burst or lyse. And you can see what happens to red blood cells. We'll look at those first. If a red blood cell is placed in a hypotonic solution like distilled water, water will enter the cell and it will lyse. It will burst. If the red blood cell is placed in an isotonic solution, like they do when they give someone fluids at the hospital, when someone has to get fluids through an IV, those fluids have a normal concentration of salt or normal saline, which is 0.89% salt, because that is the same concentration as inside of our cells. And by doing that, the red blood cells will not be affected. We will not have any lysing or any shrinking of red blood cells. But if you place red blood cells in a hypertonic solution that has a higher concentration of solute than inside the cell, water is going to leave the cell and the cell is going to shrivel. And there's actually a word for that. It's called crenation. Now, it's different in a plant cell because a plant cell has a cell wall. So in a hypotonic solution, the plant cell will take on water, but the cell wall will keep it from bursting. So we say that it has turgor pressure. It is turgid. Then if we put a plant cell in an isotonic solution, it will remain the same size. But if we put it in a hypertonic solution, even though the cell wall doesn't change, you can see the plasma membrane is pulling away from the cell wall, because inside that cell is actually losing water and shrinking. It's just that the cell wall stays the same size. I think I mentioned this in the last chapter, but that organism that we looked at a picture of on the first page of the chapter 6 slides, it was a paramecium. Remember, it had it was a single-celled protozoan that lives in pond water, and it's covered in cilia. And because it lives in fresh water, it's always taking on water. So it has a contractile vacuole that pumps excess water out of the cell to help it adjust to the fact that it's in a hypotonic situation. Okay, so here's a picture of the paramecium, and you can see the contractile vacuole. There's a video of living paramecium under a light microscope. This would be kind of cool to watch. This is in section 7.3. And I have already covered all of this. You can look at Elodea plant cells placed into hypotonic solution and becoming turgid. There is a word that I want you to know, and it's called plasmolysis. When plant cells are placed in hypertonic environments, such as like salt water, like a high concentration of salt water, then when the plasma membrane pulls away because the cell is shrinking, we call that plasmolysis. It causes the plant to wilt, and it could die from that. And you can watch plasmolysis in this video. This is actually what you would see if you were looking in a microscope. So I like these videos. Now we're going to talk about facilitated diffusion. And that just means it's still diffusion. It's still passive transport. Molecules are moving from their high concentration to their low concentration. But a carrier or a channel protein is used. Okay, so facilitated diffusion uses a channel or a carrier protein. So that means that the substances moving across the plasma membrane are either polar or they have a charge, they're ions. And you can see here that this substance is this little red triangle, has a higher concentration in the extracellular fluid. So by diffusion, it would want to move into the cell. And it's going to do that through a carrier protein. And so that's what we call facilitated diffusion. And you can watch this video as well in section 7.3. Aquaporins are involved in the facilitated diffusion of water, and ion channels facilitate the diffusion of ions which have a charge. This is showing a solute molecule diffusing through a channel protein. Some ion channels are gated channels. That means there's a gate that closes that channel and it opens in response, for example, to a nerve impulse or a chemical binding to the gate. Carrier proteins are similar to channel proteins, but they're going to change shape. So you can see that happening here. The solute molecule is the blue round molecule, and its concentration is high outside the cell. So it's going to bind with the carrier protein, cause that carrier protein to change shape, open up, and release it into the cell where its concentration is lower. Now, facilitated diffusion is a type of passive transport, but there are molecules that are transported against their concentration gradients, and that is called active transport. Active transport requires energy, so typically that's in the form of ATP, and it moves substances against their concentration gradient. So that means from their low concentration to their high concentration. And the proteins that are involved are carrier proteins. It's always carrier proteins that are involved in active transport. So what we're going to look at is the active transport of sodium and potassium. And that carrier protein is called the sodium-potassium pump. And what it does is it transports 3 sodium ions out of the cell for every 2 potassium ions into the cell. The concentration of sodium is high outside the cell, so it's active transport because it's the sodium-potassium pump is still pumping sodium out even though its concentration is high. And same thing with potassium. The potassium concentration is high inside the cell, but the sodium-potassium pump still pumps potassium into the cell, even though its concentration is high. And you can watch this in a video. 3 sodium ions are pumped out for every 2 potassium ions pumped into the cell. And this video, section 7.3 again. And we've got a review of passive and active transport. We've got diffusion of molecules directly through the phospholipid bilayer from their high to their low concentration. The only molecules that can do this are going to be nonpolar. That means because they are soluble in lipids and not water. And they're also going to be very small. So carbon dioxide and oxygen can diffuse directly through the phospholipid bilayer. Then we have facilitated diffusion, where molecules move from their high to their low concentration through a channel or a carrier protein. These molecules are always either polar or they are charged, they're ions, and that is because they are soluble in water and not lipids. Active transport uses a carrier protein to move molecules against their concentration gradient toward their high concentration. And all plasma membranes have a voltage. If you were to measure with a voltmeter, you would detect a voltage across that membrane. And instead of calling it voltage, we call it membrane potential. So that membrane potential is important for a lot of reasons, but keeping the inside of the cell having a negative charge, kind of like a battery, keeping the inside negative and the outside positive is very important for lots of different functions in cells. Okay, here is another type of active transport, which is called the proton pump. All right, so I don't know that we have talked about hydrogen ions being protons. Maybe I did, but I can't remember. But hydrogen ions, okay, so you have to understand what a hydrogen atom is. A hydrogen atom has 1 proton, 1 electron, and 0 neutrons. 1 proton and 1 electron. So when that hydrogen atom loses an electron and becomes hydrogen ion, it only has a proton. So a hydrogen ion is just a proton. So a proton pump is really pumping hydrogen ions. And by pumping, I mean it's pumping hydrogen ions toward the higher concentration of hydrogen. And that happens just to regulate pH, to regulate the acid and base concentration inside the cell. Sometimes transport occurs with 2 substances at one time, and that's called co-transport. Here is an example of a hydrogen ion sucrose co-transporter. So the proton pump keeps the concentration of hydrogen ions high outside the cell. And then when they move back into the cell by diffusion, sucrose is able to move back in as well. That's co-transport of sucrose and hydrogen ions. And we're going to stop here. I will start with this next slide, which kind of continues on with co-transport. It's going to be the co-transport of sodium and glucose.